We're going to listen to the next paper, that's granulocite signature in organ inflammation in TLR7 responsive mice is RNA and type 1 interferon dependent from Sun, Whiteman, Thiel, Agarwal, Buckler, Hammerman, Liggett, Ledbetter, Elkon from the University of Washington in the US. Please, Doctor.
Okay, sir. Okay, thank you. Good morning. This is Seattle, Washington. You see it's very quiet and peaceful. And I came to Buenos Aires, and I was told no matter how good the science, unless you do the tango, it's no good. So I have one disclosure. So as all of you know, the pathogenesis of lupus there were several landmark studies in 2003 indicating the importance of interferon alpha in lupus, and how that occurs is really a major question. Interferon, as you know, is very important because type 1 interferon activates all components of the immune system, both the adaptive and innate immunity. So there's only one mechanism that we do know of where interferon is induced in lupus, and this occurs through ingestion of immune complexes containing nuclear protein particles or ribonucleoprotein particles that get into plasmacytodendritic cells. And inside the PDC, the nucleic acid component activates the toll-like receptors, which include 3, 7, 8, and 9. So that's the only proven mechanism whereby interferon is induced in lupus so far. So because of that, we became very interested in the role of TLR7 in lupus, and there's a lot of additional evidence in both animals and humans to indicate that TLR7, which is the receptor that responds to single-stranded RNA, is important. For example, BXSB mice, one of the lupus strains of mice, have 2 copies of TLR7. There's a knock-in of TLR7 that causes lupus-like disease, and that's the mouse I'm going to be talking about a lot this morning. And then knockout. So if you knock out the TLR7, that protects MRL-LPR mice from lupus. And then in humans, there's increased frequency of SNPs in TLR7 encoded genes in males with lupus, and the interferon signature is much higher in patients who have autoantibodies to ribonucleoprotein containing antigens. So a lot of evidence implicating TLR7 in lupus. Okay, so a lot of the original work done on inducing interferon in lupus was done by Lars Ronblom, and he showed that you could actually produce this in the test tube. So, for example, if you take lupus serum and you expose it to expose it just to normal cells that contain PDCs, plasmacytodendritic cells, those cells will make interferon. So you just add lupus serum to normal cells and they make interferon. Now, if you do the same thing, but this time you add RNase to degrade the RNA that's activating the toll-like receptor, you prevent interferon from being produced. So if you add RNase, then you prevent the production of interferon because you're degrading the nucleic acid component of the immune complex. So we thought if the nucleic acid component of the immune complex is so important in stimulating interferon, could we do a proof of principle experiment in a mouse? So what we did is to make a transgenic mouse that overexpresses RNase. Okay, so we made a mouse that overexpresses RNase, and then the idea is to cross that to a lupus-prone strain of mice, and then see if we can prevent the lupus. So basically, this is just showing you quickly the production of the transgenic mouse that overexpresses RNase. So we put that on an albumin promoter. I show you here that this mouse produced RNase. So we're measuring this immunologically by ELISA, and we're also measuring it functionally by a single radial enzyme diffusion assay. And so this mouse is producing RNase. And the mouse is essentially normal. I won't go through all the details, but basically the mouse is normal. There's no adverse effect of overexpressing RNase in these animals. The immune system was normal. So then we crossed the RNase transgenic mouse to the TLR7 transgenic mouse. So now we're making a double transgenic mouse. We got the RNase overexpressor. We're crossing it to this TLR7 transgenic mouse that develops a lupus-like disease. And it also develops anti-RNA antibodies. And just to briefly summarize, this mouse was made by Sylvia Boland at the NIH. And essentially, it has a lot of lupus-like features, including antibodies that actually mostly react with cytoplasm and nucleoli. And there's several kinds of these mice, and they die early from various... It was said that they would die from glomerulonephritis. So here's the RNase expression in these double transgenic mice. You can see it's the same as a single transgenic mouse. Here we just show you that the enzyme is functional and active in these double transgenic mice. And here's the interesting part. So we wanted to see, obviously, whether the overexpression of the RNase would make these lupus mice better. And as you can see in this slide, here's the survival of the TLR7 transgenic mouse by itself, and here's the survival of the double transgenic mouse, the mouse that's overexpressing RNase. So RNase is protecting these mice from early death. And here you can see that the immune system improves significantly. There's reduced expression of activation. There's a reduction in the size of the spleen here. And there's reduced expression of activation markers on T cells and B cells. And in the kidneys, although there is still some immune deposition in a double transgenic mouse, it's much less than in the single transgenic mouse. So this reduced deposition of immune complex in the kidney. So the end of this part is that there's the degradation of the TLR7 ligand, that is destruction of the RNA by RNase, reduces immune activation, and the glomerulonephritis gets somewhat better. But the important thing we noticed when we really studied this mouse in some detail is that these mice did not have very bad kidney disease. They actually have very little proteinuria. So we figured because their survival was improved, there was something else interesting going on. And what we found in these mice was that they have very severe liver disease. They have massive inflammatory infiltrates. There's a lot of myeloid infiltration in the liver. And we think that actually is the major cause of mortality, because we can find a lot of, when we stain by caspase-3, activated caspase-3, we find a lot of cell death in the single transgenic mice. And in the double transgenic, there's a marked improvement in the liver inflammation and liver function. So the other interesting thing we found in these mice is that they have a marked expansion of their myeloid compartment, the inflammatory monocytes. And the interesting thing, if any of you went to the talk yesterday on the nets by Dr. Kaplan, you know that these myeloid cells, the neutrophils, have a lot of enzymes, granulocite enzyme, proteinase 3, and so on, that are very characteristic of the activated cells in lupus patients. And these monocytes, inflammatory monocytes, have enormous upregulation of these scale. This is a 100-fold elevation of expression of myeloperoxidase, proteinase 3, and elastase in their inflammatory monocytes. And so if you stimulate these cells with TLR7 ligand, they produce a lot of TNF-alpha, and this is just summarized in this bottom panel here. There is a marked increase in production of TNF-alpha after stimulation of these cells with the TLR7 ligand. So what we conclude from this part is that the TLR7 transgenic mice die not from kidney disease, but they're dying from severe inflammatory disease in the liver. They have a myeloid expansion, and these activated myeloid cells, which are like a very immature neutrophil, have a huge expression of proteases that probably contribute to the disease. And these monocytes are hyperresponsive to TLR7 ligand, and they make a lot of TNF-alpha. So the questions that we're busy studying right now are, what is the ligand that activates the TLR7? And how does that get activated in the animal? And what is the mechanism of liver cell death? Is that through TNF? And then the question is, to what extent do the inflammatory monocytes resemble these low-density granulocytes that were discussed yesterday by Dr. Kaplan and initially reported by Dr. Pasquale. And all of this we think is actually highly relevant to human lupus, where, as I told you, there's good evidence that the TLR7 is activated and may be contributing to disease pathogenesis. And because of that, we have made a human biologic. Jeff Ledbetter, who many of you know made Orencia, has helped us to make a human biologic comprising IgG and RNAs that are in safety studies now, and we hope one day will be tested to see if we can treat human disease. So that's all just to mention people at the University of Washington who contributed to these studies, and thank you very much.